welcome to another episode of This Week in IP&O. We are, of course, as always, live from the table in my kitchen. We have a very special guest. We have Dr. Dina Fonseca, who is a professor of entomology here at Rutgers. Thank you for joining us and coming on the show.
Having me. I'm curious to know how this is going to go. So let's do it.
You know, we're an easy bunch to get along with. We're just going to keep a little conversation flowing.
Okay.
So I guess just to start us off, can you just tell us a little bit about your role at the university and what entomology is for people that might not have heard of that word before?
Okay. So, well, entomology is the study of organisms that have articulated body parts, things like insects. You know, in many ways, entomology is in large, because there's so many insects, is about insects. But we also work on ticks and things like spiders, which are not insects. So they're arachnids. So mostly what I do, so entomology is a broad field. It includes the study of insects and arachnids, but my specific specialty is those that have a medical or a veterinary importance. So that's why I gave the examples of mosquitoes and ticks and to some extent spiders. So I work with organisms that can do damage to animals. So I have colleagues in our entomology department that are working on agricultural pests, something like spotted lanternfly. And we all find things like cicadas really interesting. But in terms of the kind of research we do, we are mostly interested in, again, like I said, things that can really hurt us. And we admire them as much as we want to some extent, trying to limit their damage. I'm a professor of entomology. I'm also the director of the Center for Vector Biology, which is an extension program that works primarily with the mosquito control programs across New Jersey. There's 21 counties, 21 mosquito control programs. Those are our primary stakeholders, but we also work directly with the public and also increasingly with the increasing number of professional pest managers or pest companies that are starting to work on mosquitoes and ticks. So sort of that's it in a nutshell.
So we've heard about the citizen project called New Jersey Tick Map that you've been working on. Can you tell us a little more about that project? So,
I mean, you guys are like, this is like hot off the press because we just very recently obtained funding from the New Jersey Assembly. It was actually in the budget, in the governor's budget, a line item to develop tick research and control. Ticks are a really significant problem across the Northeast New Jersey at the epicenter of cases of Lyme disease. I mean, there are about 40,000 cases of Lyme disease every year in the United States, and 95% of them are in the Northeastern United States and also in the Midwest, something like Michigan and Wisconsin and Minnesota. So we have, I'm sure many of you have either had Lyme disease or know somebody that had Lyme disease. So ticks are becoming very important, but there's actually there has been very little research on ticks, by their biology, and in particular, how to survey and control them. This is, you'd be surprised to know this, but there we go. So again, we have about a year to demonstrate what we can do across the entire state of New Jersey. This is created by the Assembly to try to both provide information on the populations of ticks across the state, and also to some extent have people understand a little bit more what ticks are and how they can protect themselves from ticks. I mean, there's a much better understanding of mosquitoes than ticks. Ticks are sort of mysterious. Often people don't even know what they look like. And then when I say people, I'm actually including also medical doctors. There are cases, not in the United States, but there have been cases of people thinking that a tick embedded is actually some sort of, wart or a cancer, and have actually done surgical interventions when they simply could have just popped it out. So we decided, it's again, brand new project. We're just starting, and this is going to heat up, and you guys have it first. The New Jersey tick map. We're going to be a large citizen science project. We're going to invite people to send us any ticks they find on themselves, on the environment around their houses, and submit it to us. We'll immediately provide them information on the identification, and then we'll use those ticks to a broad scale analysis of tick-borne pathogens like Lyme disease and others. There's multiple pathogens that can be transmitted by ticks, including a deadly one, Powassan virus, that just recently actually killed a New Jersey resident. So this is a significant problem, and that's basically the extent. Now we're hiring people, trying to develop a web page, and trying to send the message out, and here we are. Message started.
So I have this kind of simple A or B. You could tell me if this is correct. Deer ticks are the bad ones. Dog ticks are the less bad ones. Is that correct?
Well, okay. So first of all, deer ticks is not the accepted common name for these ticks.
Okay, yeah.
It's kind of a minor problem, but which is, they're called black-legged ticks, Ixodes scapularis. Black-legged ticks are in fact the primary bad ones. They do transmit Lyme disease, and only they can transmit the Lyme bacterium, the bacteria. They also transmit anaplasma, another bacteria. They're the ones who transmit Powassan, and they transmit a protozoan, a little similar to malaria, called Babesia. And so, yes, black-legged ticks, which are primarily forest ticks, are the primary guys. The dog tick are much larger, and they are not considered at this point. They have been associated with a really nasty bacteria called the Rocky Mountain spotted fever, or Rocky Mountain spotted bacteria. And so it depends really who you're referring to. Rocky Mountain spotted fever bacterium can kill you. But we've actually done a recent publication, actually, one of my students, and shown that in New Jersey, the dog ticks are not infected with this bacterium. And it's a confusing and sort of, it has come from again, a limited amount of research on these ticks to have this idea that dog ticks can transmit Rocky Mountain spotted fever. We have found no evidence of that. And we've done extensive surveys across New Jersey. So yeah, big one, probably less likely to be a problem. Tiny little guys, the little black-legged ticks, AKA the deer ticks, they look like a triangle, tiny little black triangle. And it's actually the nymphs that are at their highest numbers in April, May, June, which is why May is a Lyme awareness month. Those are the ones who are more likely to actually be the problem, because they're small. And one thing I got to explain is that, so ticks have 4 stages. They have eggs, the larvae, which only have 3 pairs of legs, the nymphs, which have 4 pairs of legs, slightly bigger, and then the adults, which also have 4 pairs of legs. And again, each stage is a little bigger. The larvae are primarily, when they're born, they're not infected with anything. This is true in large measure of most vectors. When they're born, they're not infected. Then they take a blood meal. And in the ticks, they only feed on blood. They don't feed on anything else. So they need to take a blood meal from the larva to become the nymph, and then the nymph to become the adult. And every time they take a blood meal, they increase the likelihood of being infected with a pathogen, because they're picking up the pathogen, the virus, the bacteria, from their hosts. So you'd think that the most dangerous stage would be the adult, because it's taken 2 blood meals, so it's more likely to be infected. But adults are mostly active in the fall during the deer rut. So, and by the way, these ticks are active all year round. In January, in a beautiful sunny day, you can get a bite from a black-legged tick. So you'd think those guys would be most dangerous, but actually they occur at a time when people have long sleeves and boots and are in the field, kind of like protected from the cold, while the nymphs, which are smaller, are showing up in the spring when you're like, yay, shorts, I'm going outside. And that's when you end up, and they're much, much smaller. So they're much harder to detect. And sorry, I have to do this sort of PSA in here. the best way to control, to not be affected by tick-borne diseases from the black-legged tick is to do tick checks. And so if you're going to be outside, go indoors, have a shower, and then check yourself out, have somebody check out parts you can't see. And really, it's a nice bonding experience, and you really decrease the likelihood of getting Lyme disease or other tick-borne diseases. So again, they're really, really small. And this is something we're trying to do for the New Jersey tick map, is provide information and like embedded little blocks with different stages of ticks, so that people can really understand what it is that they're looking for. And that is a huge step in terms of prevention. Ticks take several hours to start feeding on you. So they're not like a mosquito. Mosquito is like time and they're done, right? With ticks, they go on you, they look for a place to feed. It usually takes them like a day to finally make decisions, and then takes them another few hours, up to a day, to start transmitting the pathogens that they're infected with. So there is a window of opportunity, and that's why tick checks are a really good way to decrease that. So there, lots of information.
Yeah, no, that's great. Is it primarily deers that they're picking this stuff up from, or is there
no, they're feeding on. This is why the deer tick is a misnomer. Deer are very important hosts of the adults. And in fact, deer actually clear the pathogen from the adults. The immune system of a deer, when the tick is feeding on a deer, which is already the adult, basically all that's going to happen now is going to lay eggs. It's not going to bite anybody else.
Yeah.
They're actually picking up the pathogens from white-footed mice. The little mice is what the larvae and the nymphs are mostly feeding on. And so those are the ones that are the reservoirs of most of these pathogens. So it's really controlling the mice, and controlling the infection on the mice is a better way to control the disease. Decreasing the population of deer is a better way to control the ticks. So if you're trying to prevent ticks from laying, they lay 3,000 eggs each female. So if you prevent them from feeding on deer, then you're preventing the next generation from being, preventing a large number of ticks from joining the next generation. So it's just two different control measures: controlling the pathogen versus controlling the tick populations themselves.
Oh, great. Thank you for that.
Switching topics a little bit. We were wondering what your plans were for utilizing the Rutgers Garden in some of your research.
Yeah, so we've actually already using the Rutgers Garden. So we're doing surveys of mammals on Rutgers campus, on both Douglas and Cook. We started in the spring. And again, just completely linked to the question before, by collecting the mammals, you can look at, so you basically set up these traps. We've been setting up the traps at University Inn, at the Rutgers Gardens, and also in the goat farm off the College Farm Road, the poultry farm area. So we set up these traps, and we collecting both small mammals, so mice, voles, chipmunks, but also medium-sized mammals like raccoons and skunks. Okay, we got skunked a couple of times. It's a dangerous profession. Oh, no. So skunks and groundhogs. We had if you may remember, there were some a black bear at some point in the Rutgers Gardens. So one of my graduate students got a text saying, "Hey, there's a bear behind you," and everybody's okay, except one of the traps got kind of mangled by the bear, like literally just bent. Oh, wow. Probably had a mice inside, so it was like a Twinkie for the bear. So we've been doing these trappings already using the grounds, and the Rutgers campus is quite well endowed in medium and small and medium-sized mammals, and lots of deer. There are lots of deer around. So then we started thinking about, like, by experiencing the gardens in particular and University Inn, we thought we could engage the public in ways to try to assess how different interventions for controlling ticks may be working. So we're thinking of creating a small kit, something like a Ziploc bag with a map. University Inn already has an app, and we're trying to work with them to do this, where we would have treated areas like in the gardens, just beautiful areas, and it would treat the paths. When people are walking through the gardens, they're actually picking up ticks from the sides of these paths, either from sort of short vegetation or a bit taller vegetation. And so we would have information, and then we provide them with double-sided tape. They would put on the base of their pants. And then we would engage these citizen volunteers, researchers, to help us by keeping track of where they've been, where they've walked, by putting on the map. And at the end, just give us back, just take the tape, fold it, put it in a Ziploc bag, and leave that at the entry of the grounds. And then we would be able to use that information to see if the interventions we did in some of the areas limited the number of ticks that people picked up. So trying to do these are complicated experiments because they only work at large scale. They're so-called area-wide. You have large areas that you have to treat, and then you have to figure out ways of sampling and examining them. And ticks are sampled by simply dragging a piece of cloth over the vegetation. It's very time intensive, and to be honest, I mean, getting the information from the people, like what did people pick up, is actually ultimately what we want to know. We want to know if we're protecting the visitors, and so it's actually the best measure of effectiveness. So that's what we're planning on doing. You can see lots of ideas, and can use any help you can provide. So that's the idea, to use visitors as experimental subjects and also as technicians collecting data for us.
I have one more question that's kind of a little off script, but I feel like every summer I'll be like sitting outside with 2 or 3 people, and at the end of the night I feel like I get like eaten alive by mosquitoes, and everyone else is fine. So is there something about like specific people that make them more prone to getting bit by mosquitoes, or is it just in my head and I'm just like taking this situation and applying it to others?
Okay, so I get that question every single time. Every single time. And the answer is twofold. And I don't have an actual complete answer because it's actually extreme. So let me start with the bad news. It's extremely hard to get funding to address that kind of question. It's not a simple question. It requires, ways of sampling people, which is complicated. It involves potentially exposing people to mosquitoes, which is complicated. And it doesn't necessarily make much of a difference. Like just because you're getting bit, as long as you're not getting sick, the NIH, National Institutes of Health, be like, we gotta fund something else. There's a couple of papers that came out a few years back that actually concluded that your blood type made a difference. So if you were a blood type A, you're less likely to be bit. And if you're blood type O, you're more likely to be bit. And the other blood types were somewhere in the middle. These were two studies actually related. They actually did a really good job of trying to go around some of the problems of variability among people. But it's pretty limited sample size because, as I said, it's really hard to do work on people, especially because you have to expose them to the risk of being bit by mosquitoes. And again, it's a complicated thing to do. There's also a study, actually, there's someone at Princeton University that has been, Lindy McBride, that has been looking at, she started out by trying to understand more like the evolution of the mosquito. And she was basically, her hand was basically what she used as the human. And she wasn't getting bit, it was just looking at these olfactometers, looking at which way the mosquitoes were going, to a person or to a hamster. She was looking at the evolution of mosquitoes biting people. And then she got her own lab, got her own postdoc, and had the postdoc do these kinds of studies too, and things were not working right. And she realized that there was this one chemical, sulcatone, that varies quite a bit. It's basically our skin, our microbiome is basically producing the smells that mosquitoes are using for making decisions about biting or not biting. They retract it from CO2, the amount of CO2 I'm producing. If I've been running, if I'm a particularly large person, I'm going to be producing more. So they come towards you. But then in terms of making decisions about who's, like if you're in a group of people, like you said, you're all producing CO2. And then making a decision about which one of you the mosquito is going to bite is going to be determined most likely by your microbiome and by characteristics of your skin. And maybe by your blood group. It's a little, that was a couple of papers, that's all I can offer, but that's basically it. Now, that's the other side of it is that some people have higher, stronger reactions than others. So your immune system could also be part of the reason you may notice more. You may get a welt, you may get itchy, while somebody else that actually may be getting the same exact number of bites that you are, just not noticing it. So as I said, it's really a complicated, it sounds so simple. Why am I the one who always gets bit by mosquitoes? Why is it, X never gets bit by mosquitoes? But the fact is that it is a complicated answer that is going to come up eventually from that one question. And not a lot of funding agencies are especially convinced that we need to spend a lot of money on that, as opposed to trying to come up with strategies for prevention or other things.
All right. Well, thank you for coming on the show, and thank you for teaching us a little bit about what you do. And we will, as always, give you the last word if there's anything else you'd like to say about the new tick program or about anything you want.
Well, I teach medical and veterinary entomology, so any in the audience that want to take a class that is that makes you think, that we talk about Black Death and yellow fever epidemics and all that fun stuff. Again, the only other thing I could say is that the New Jersey tick map is just starting. We're creating this, so don't be surprised you don't hear about it for a bit. Probably it'll really show up in your podcasts or maybe in your feed more in the spring. But we're going to try, and if you're interested in collaborating, just contact me, and we'll put you to work. There's lots to be done. Thank you for this opportunity.
Thank you.
Thank you.
Thanks everyone for listening.